Welcome back to the deep dive. So if you think about the nervous system, it's like the body's super fast fiber optic
Yeah.
network, right? Instant messages.
Yeah, exactly.
Then the endocrine system, well, it's more like the global postal service.
Mm.
Slower, maybe, but steady. And it gets messages to, well, basically every target cell eventually.
That's a great way to put it.
And our mission today is really to crack that chemical code.
Yeah.
We want to uncover the basics, the mechanisms behind how hormones work, how they travel, and, the command center that tries to keep it all in balance.
And that global network idea is spot on because we really have to think beyond just the classic glands like the thyroid or adrenals.
Yeah.
It's this deeply integrated system. It's involved in growth, metabolism, differentiation, and maybe most importantly, adaptation, how the body adjusts.
Okay.
And what's really fascinating, I think, is that it's not working in isolation. The endocrine system is constantly talking with the nervous system and the immune system. It's like a three-way chat we call the neuroendocrine immune interaction.
Okay, let's unpack that a bit. So the basic unit here is the hormone. I find it interesting that the name actually comes from a Greek word meaning aroused to activity. Because they aren't, doing the work themselves. They're the messengers telling the cells what to do or when to do it. They're these specialized organic molecules floating around modulating responses.
Exactly. They're signaling molecules. And they don't just come from the places you'd expect. You mentioned integration. Think about the heart, for example. It's the main source of atrial natriuretic peptide, ANP.
The heart makes hormones.
Yeah. And that ANP travels all the way to the kidney to have its effect. Or think about the kidney itself. It makes erythropoietin, EPO, which then goes through the blood to the bone marrow to stimulate red blood cell production.
Wow. So organs we think of doing one job are actually part of this chemical network, too.
Absolutely. They're active endocrine players.
But wait, if the heart and kidney are making hormones, why do we still just call it the endocrine system? Shouldn't it be, I don't know, the organ messenger system or something?
That's a fair question. It's still endocrine because the defining feature is how the hormone acts. Even from the heart or kidney, it's released into the bloodstream. That's the endo part, meaning within tried to travel and act on a distant target, the crine or secretion part.
Okay. So it's about the journey.
Precisely. And that journey, or rather how far the message travels, defines the different types of hormonal action. It's not all long-distance mail.
Right. It's not one size fits all. So if we stick with the communication analogy, what are the different delivery methods? You mentioned 5.
Yeah, 5 main ways. First, you've got the classic endocrine action. That's your international airmail. The hormone gets into the blood, travels a fair distance to reach its target cells, like that AMP going from the heart to the kidney.
Okay, the long haul.
Then you narrow it down. Paracrine action is more like talking to your next-door neighbor. The hormone acts locally on cells right near where it was released. Crucially, it doesn't get into the bloodstream. Think of sex steroids acting right there within the ovary.
Local gossip, basically.
Kind of. Then it gets even more personal. Autocrine action is like sending a memo to yourself. The hormone affects the very same cell that produced it. Insulin, for example, can actually inhibit its own release from pancreatic beta cells.
Self-regulation.
Exactly. And even more contained is intracrine action. Here, the hormone is made and acts inside the same cell without ever leaving.
Okay, so that's 4. What's the 5th? The special delivery.
That would be the neuroendocrine effect. This one starts in the nervous system and neuron makes and releases the hormone. But then instead of acting like a neurotransmitter across a synapse, it gets dumped into the blood and travels just like a regular endocrine hormone.
So it crosses systems.
Right. Think of antidiuretic hormone, ADH, or even epinephrine released from the adrenal medulla, which originates from neural tissue, neural source, bloodstream delivery.
Got it. And I guess the type of delivery influences the type of package, right? The structure of the hormone itself.
Absolutely. The structure is key. We generally group them into 4 types. You've got the relatively simple amines and amino acid derivatives like epinephrine or thyroid hormone.
Okay.
Then the big family, most hormones actually are peptides, proteins, or glycoproteins, things like insulin, growth hormone.
Right.
Then you have the steroids, which are all derived from cholesterol. Think glucocorticoids from the adrenal cortex, or estrogens and testosterone.
Lipid-based.
Exactly. And finally, the fatty acid derivatives, like the eicosanoids, which include prostaglandins. These often act locally.
Okay, so amines, peptides, proteins, steroids, and fatty acids.
Yeah.
And you mentioned lipid. That structural difference, especially whether they dissolve in fat or water, is crucial, right? That brings us to transport and how long they last.
Precisely. That lipid solubility, or lack thereof, is the absolute defining feature when we talk about transport and half-life. Are they water-friendly or are they, oily?
So how does that work in the blood, which is mostly water?
Well, the water-soluble ones, the peptides and the catecholamines like epinephrine, they're fine. They dissolve easily in the blood, circulate freely, kind of like jumping on a bus. They get where they're going, get metabolized pretty quickly, and don't need special help.
Easy enough.
But the lipid-soluble ones, the steroids and thyroid hormones, they hate water. They're hydrophobic, so they can't just float around freely in the blood plasma. They need an escort.
Escorts.
Yeah, carrier proteins. These are usually synthesized by the liver. The hormone binds to the carrier protein, which shields it from the watery environment and carries it through the bloodstream.
Okay, that makes sense, but it sounds like an extra step. Why does the body bother with these carriers? It seems complicated.
But it serves a really important purpose. It determines persistence, how long the hormone sticks around.
How so?
The extent of protein binding is directly related to the hormone's half-life. Think of thyroxine, T4, the main thyroid hormone. It's massively protein-bound, like over 99% bound. And because it's so tightly bound, it hangs around for ages. Its half-life can be up to 6 days. It acts like a slow-release capsule, providing sustained action.
6 days. So you could miss a dose of thyroid medication and still have active hormone floating around from days before.
Exactly. It gives you that biological persistence. Now, compare that to something like angiotensin II, which is involved in blood pressure. It's water-soluble, circulates unbound.
The sprinter.
The sprinter. Its half-life is less than a minute, perfect for making rapid adjustments to blood pressure, but not for sustained effects.
That difference is huge. And that protein binding thing, that has clinical relevance too, right? Like with medications.
Oh, absolutely. This is really important. If a patient is taking a drug that also binds to the same carrier protein, say albumin, that the hormone uses,
they compete for seats on the bus.
Exactly. The drug can kick the hormone off the carrier protein. Suddenly you have more free, unbound hormone floating around, and it's the free hormone that's biologically active.
So you could inadvertently boost the hormone's effect just by adding another drug that uses the same carriers.
That's right. You increase the signal strength without actually increasing hormone production, which can cause problems. And quickly on elimination, the water-soluble ones are generally broken down quickly by enzymes in the blood or tissues and excreted by the kidneys or liver. The bound ones, the steroids and thyroid hormone, they stick around longer because the carrier protects them. They only really get eliminated once they detach and become free, or after they're metabolized, often in the liver, to become more water-soluble for excretion.
Okay, so structure dictates travel. Travel dictates lifespan and binding. Makes sense. Now here's where, for me, it gets really interesting. How the cell actually receives the message. The communication at the cellular level.
The receptor interaction?
Yeah, it's not like the cell just listens to any hormone shouting at it. It needs a specific receiver, right? A high-affinity receptor. Like a very specific lock for a very specific key.
Precisely. High affinity means it binds tightly, even when hormone concentrations are really low. And a cell's responsiveness, its ability to actually hear that hormonal message, depends on two main things.
Which are?
One, the number of receptors it has on its surface or inside, and two, the affinity or stickiness of those receptors for the hormone.
And can the cell change that? Can it adjust its hearing?
It absolutely can. This is a key adaptive mechanism. If hormone levels are chronically low for a while, the target cell can compensate through upregulation.
And upregulation sounds like turning up the volume.
Kind of. It actually increases the number of receptors, makes the cell more sensitive, trying to catch every little bit of that faint signal.
Okay. And the opposite?
The opposite is downregulation. If the cell is just bombarded with excessive hormone levels for a prolonged period, it protects itself by decreasing the number of receptors. It's trying to dampen the signal, reduce its sensitivity to that overwhelming noise.
So it's trying to maintain balance, even when the signal is too loud or too quiet. Now, the core mechanism you mentioned, receptors on the surface or inside, where that lock is located depends entirely on the key, right? Whether the hormone can get through the cell door.
Exactly that. It comes back to water versus lipid solubility. The water-soluble hormones, your peptides, your catecholamines, they're generally charged molecules. They can't just slip through the fatty cell membrane.
So they're stuck outside.
They are. So they use cell surface receptors. The hormone itself acts as the first messenger. It binds to its specific receptor on the outside of the cell, but it never actually
Okay,
enters.
so how does the message get inside?
That binding event triggers a change inside the cell, usually activating a second messenger system. Things like cyclic AMP or CAMMP are common second messengers. They get generated inside the cell in response to the hormone binding outside, and they carry out the instructions within the cell.
So the hormone just rings the doorbell, and the second messenger opens the door and does the work inside.
That's a perfect analogy. The first messenger never crosses the threshold.
But then you have the lipid-soluble crowd. The steroids, the thyroid hormones, they play by different rules.
They definitely do. Because they are lipid-soluble, they can diffuse easily right through the cell membrane. No problem getting inside.
So they don't need a doorbell or a second messenger?
Nope. They use intracellular receptors, which are often called nuclear receptors because that's where they ultimately end up doing their work. The hormone slips inside the cell, finds its receptor, usually floating in the cytoplasm or sometimes already in the nucleus.
In that.
The hormone binds to the receptor, forming a hormone receptor complex. That complex then travels into the cell nucleus, finds specific DNA sequences called hormone response elements, and binds there.
So it's interacting directly with the genes.
Correctly. By binding to those elements, it modulates gene transcription, turning genes on or off. This changes the rate of protein synthesis, effectively altering the cell's function by changing the proteins it makes. They rewrite part of the cell's instruction manual directly.
Wow, that's a fundamentally different way of operating. Cell surface with second messengers versus straight into the nucleus to mess with DNA expression. Huge difference.
It really is. Dictates the speed and duration of the response, too. Second messengers often cause rapid but maybe shorter-lived effects. Changing protein synthesis takes longer, but the effects can be much more sustained.
Okay, that paints a clear picture of the cellular action. Now let's zoom out again. We need to look at the control tower, because hormone levels aren't just constant, are they? You mentioned things vary by time of day, like growth hormone and ACPH having diurnal patterns.
Right, often peaking in the morning.
And others have cycles, like monthly cycles for sex hormones. How is all that regulated?
That regulation largely comes from the big coordinating center, the hypothalamic pituitary, or HP, functional unit. The hypothalamus, located at the base of the brain, is really where the nervous system talks to the endocrine system.
How so?
It receives all sorts of input from the brain and body signals about stress, emotion, pain, temperature, sleep cycles, you name it. And it translates those neural signals into hormonal commands.
And the pituitary gland hanging just below it is like the middle manager
Yeah.
executing those commands.
Kind of, yeah. But the pituitary itself is actually split into two very distinct parts, functionally and anatomically. You have the anterior pituitary and the posterior pituitary.
Okay, what's the difference?
Let's start with the anterior pituitary, or adenohypophysis. Think of this as a true gland, a hormone factory. It's connected to the hypothalamus indirectly through a special blood vessel network called the hypophyseal portal venous system.
A portal system.
Hook in the liver.
Similar concept, yeah. It allows the hypothalamus to release tiny amounts of its own hormones, releasing hormones like TRH or GnRH, and inhibiting hormones like somatostatin or dopamine directly into this portal blood. These travel the short distance down to the anterior pituitary and tell it which hormones to make and release into the general circulation. So the hypothalamus uses chemical signals through this special blood supply to control the anterior pituitary.
Exactly. And the anterior pituitary then produces its 6 major hormones. Growth hormone, GH, thyroid-stimulating hormone, TSH, adrenocorticotropic hormone, ACTH, follicle-stimulating hormone, FSH, luteinizing hormone, LH, and prolactin.
Okay, that's the anterior part. What about the posterior?
The posterior pituitary, or neurohypophysis, is completely different. It's not really a gland that makes hormones. It's more like an extension of the hypothalamus itself.
How is it connected then?
It's connected by nerve axons. The cell bodies of these neurons are up in the hypothalamus, and their long axons run down a stalk and end in the posterior pituitary.
So direct neural connection.
Direct neural connection.
Yeah.
And the 2 main hormones released from the posterior pituitary, antidiuretic hormone, ADH, and oxytocin, they actually synthesized up in the hypothalamus in those neuron cell bodies. Then they travel down the axons, inside vesicles, and are simply stored in the nerve terminals within the posterior pituitary. When the hypothalamus decides to release them, it just sends a nerve signal down those axons, causing the stored hormone to be released directly into the bloodstream from the posterior pituitary.
So the posterior pituitary is more like a storage warehouse and release point for hormones made in the hypothalamus.
Precisely. Chemical control via portal blood for the anterior, direct neural control and storage for the posterior. It's a key distinction.
That clarifies the command structure nicely. And this whole complex system, hypothalamus telling pituitary telling target glands, it's all held in check by feedback, right?
Overwhelmingly, yes. Feedback regulation is the cornerstone of keeping hormone levels within their normal, narrow range. And the most common type, by far, is negative feedback.
The thermostat analogy again.
Exactly like a thermostat. When the level of a particular hormone gets high enough, or when the effect it produces reaches a certain point, that signals back usually to the hypothalamus and/or the pituitary to inhibit further secretion of the stimulating hormones.
So if thyroid hormone levels rise, they tell the pituitary to make less TSH and the hypothalamus to make less TRH.
Perfect example. That rising thyroid hormone shuts off its own stimulus. It maintains the set point, prevents levels from getting too high. That's the HP target gland axis in action.
Makes sense. Keeps things stable. But you mentioned it's mostly negative feedback. What's the other kind?
The other, much rarer mechanism is positive feedback.
Which sounds like it would do the opposite, make things unstable.
It can be destabilizing, which is why it's rare and usually part of a process that needs to reach a specific endpoint or climax. In positive feedback, rising levels of a hormone actually stimulate more secretion of that hormone, or the hormones that stimulate it.
So it accelerates things.
It accelerates the process. The classic example is during the menstrual cycle. As the ovarian follicle grows, it produces more and more estradiol. For a period, that rising estradiol actually stimulates the pituitary to release even more FSH and especially LH.
Leading to the LH surge.
Exactly. That surge is necessary for ovulation to occur. Once ovulation happens, the feedback switches back. But for that specific event, positive feedback drives the system rapidly towards the necessary peak. It amplifies the signal until the job is done.
Okay, negative for stability, positive for pushing towards a specific event.
Got it. So we've kind of built the whole system now. The messengers, the delivery routes, the receivers, the control center, the feedback loops. How do clinicians actually figure out if something's gone wrong? How do they test these endocrine functions? That brings us to diagnostics, right?
Right. And the tests used really mirror the physiological mechanisms we've been talking about. They're designed to probe those pathways.
So how do they work?
Well, if a clinician suspects hypofunction, meaning a gland isn't active enough, it's underproducing, they'll often use a stimulation test, trying to kickstart it.
Pretty much. You give the patient a dose of the tropic hormone that normally stimulates that gland. For example, you might give synthetic ACTH and then measure the adrenal cortex's cortisol response. If the adrenal gland is healthy, cortisol levels should rise significantly. If they don't,
Then the gland itself might be the problem.
exactly. You're testing the target gland's ability to respond. Now, if you suspect the opposite hyperfunction, or maybe a gland that's gone rogue and is overproducing hormones independently of normal control, what we call autonomy.
Then you want to see if you can shut it down.
Precisely. You use a suppression test. You administer a substance, often a synthetic hormone, that should trigger negative feedback and suppress the gland's output. For instance, giving a potent steroid like dexamethasone should suppress the pituitary's ACTH release and therefore the adrenal's cortisol production, if cortisol levels don't fall.
It suggests the gland isn't listening to the feedback signals. It's running wild.
Exactly, it's autonomous. So stimulation tests check if it can turn on. Suppression tests check if it can turn off.
Makes sense. And besides these dynamic tests,
Of course, there are routine blood tests. Measuring the actual hormone levels using very sensitive and specific methods like ELISA, enzyme-linked immunosorbent assay, is fundamental. And sometimes measuring a physiologic indicator is just as important, like checking blood glucose levels to assess insulin function, rather than just insulin levels themselves.
right, measuring the effect. What about looking at the glands themselves? Imaging.
Imaging is crucial too. MRI, magnetic resonance imaging, is great for soft tissue detail, like looking at the pituitary gland, and it doesn't use ionizing radiation. CT, computed tomography, is often faster and better for visualizing calcified structures or bone involvement, but it does use X-rays and sometimes contrast dye is needed, which has its own considerations.
Is there anything that combines structure and function?
Yes, and that's where things get really advanced.
Yeah.
PTCT fusion scanning is incredibly powerful.
PTCT. How does that work?
It overlays two types of scans. The CT gives you that detailed anatomical map of the structure. The PE, positron emission tomography scan, uses a small amount of a radioactive tracer, often attached to glucose, to show metabolic activity.
So it shows you which tissues are working hard.
Exactly. It shows you tissue function. So you can see a lump on the CT, and then the PET tells you if that lump is metabolically active, like a functioning tumor, or if it's just scar tissue, for example. It integrates structure and function beautifully.
That sounds incredibly useful for pinpointing problems. Okay, this has been a fantastic deep dive, really laying the groundwork. We've covered the chemical language of hormones, how they get around, those specific lock and key receptor mechanisms, and then the vital regulatory role of that hypothalamic pituitary axis in the feedback loops, keeping it all in check.
It really forms the foundation for understanding almost any endocrine disorder, and that knowledge connects directly to clinical situations. Like, think about a really common scenario. A patient is prescribed exogenous corticosteroids, say, prednisone for inflammation. What happens to their own natural hormone levels?
Okay, based on what we said, the body detects all this extra corticosteroid coming in from the pills.
Right,
So that triggers negative feedback.
right.
A strong signal goes back up to the pituitary and the hypothalamus,
Telling them what?
telling them, hey, we've got way too much cortisol activity down here. Shut off the stimulus. So the pituitary cuts back on ACTH production, and the hypothalamus cuts back on CRH.
Precisely. Their natural ACTH and cortisol levels will plummet. The body's own HP adrenal axis shuts down because it senses the high levels from the external source.
It's the negative feedback loop doing exactly what it's supposed to do, trying to restore balance, even though the imbalance came from medication.
Exactly. It shows just how sensitive and responsive that negative feedback system is, constantly striving for that equilibrium, that set point, even when faced with external hormonal influences. It highlights the power and the importance of these control mechanisms we've just discussed.
A perfect illustration to wrap things up. Thank you so much for walking us through all of that. It's been incredibly insightful. We really hope you, our listeners, can use this deep dive into the mechanisms of endocrine control as that solid foundation for understanding how chemical communication governs so much of our physiology.